We are pleased to welcome you to this keynote lecture on diploidization, polyploidy, and evolution of plant diversity by Dr. Michael Barker. Mike Barker is an associate professor and associate department head of the Department of Ecology and Evolutionary Biology at the University of Arizona in Tucson, US. He has studied the origins of biological diversity, particularly for the abrupt changes of genomes and some events like polyploidy, genome duplication, chromosome change, and hybridization that have contributed to evolutionary diversity of plants. A major focus of his work is understanding the evolutionary cycles of polyploidy and diploidization across the plant phylogeny. He integrates new computational and evolutionary genomic tools with traditional approaches, such as molecular evolution, phylogenetics, mathematical modeling, and experimental work to better understand the origins of biological diversity. Researchers in his lab have worked with different study systems, such as Brassicacea or Selaginella, have analyzed genomic plasticity in response to environmental variables, and paleopolyploidy across the tree of life. He has published near 100 papers and has helped develop tools for inferring paleopolyploidy, studying chromosomal evolution at the population genetics of polyploidy species, including chromEvol maps and polyploidy models for DADI. He is, I can say, very proud, and he likes very much his students as postdoc. He has so far mentored 10 graduate students and 11 postdoctoral fellows who have gone on their own great careers. Many of you probably know very well Mike Barker because he has been the organizer of the Polyploidy webinars, which ran from 2020 on Wednesdays on his website, and is going to be continued this fall. This seminar, which brought together many attendees, showcased the research of young and experienced researchers and were fantastic opportunities to learn how polyploidy evolved and the impact of polyploidy for many different perspectives and through various approaches in plants and animals. So thank you very much, Mike, for being in this 20th IBC conference and for giving this lecture.
Thank you. All right, well, yeah, thank you, and thank you all for coming out this morning. I'd also like to thank Gonzalo and all the organizers. I sort of think of the IBC as like the Olympics of botany, and they had a short window to get this one organized, and I think it's been a really fantastic success so far. And I've really enjoyed my time here in Madrid and in Spain. And I also just want to say that my very first meeting ever of any conference, not just IBC, but any botany meeting or any scientific conference was the IBC in 1999. And it was a really fun meeting, right? These IBCs are really important. And this one I remember, well, it was my first one. But I remember meeting Stebbins at this meeting, and I remember talking to him about polyploidy, and I remember seeing this whole series, and many of you were there, on Amberella being discovered as the sister lineage to the rest of the flowering plants. It just had a great time, and it certainly propelled my own enthusiasm. And personally, it's a really, just a fantastic honor to be asked 25 years later, which also makes me feel a little old now, but to come and speak to everyone about work in my lab and things that were really inspired by that first meeting back in St. Louis and all the other botany meetings and IBCs since then. And for many years, I've been really excited about polyploidy. Back at IBC in 1999, I gave a talk or a little poster on polyploidy, and back then it was a little bit like, you know, that's a nice thing, but people weren't quite as engaged and excited about it as they are now, I think. But we've known about polyploid speciation for a long time. There are many ancient or old examples of polyploid complexes and research on these complexes going back to the early days of the 20th century. For example, arguably one of the more well-known and well-worked-out cases is the Triangle of Yu by Wu Zhongjun, and this is a classic example of the allotetraploids and their diploid parents in Brassica, where you get allotetraploids hybrids between each of those diploid species in the brassicas. These are familiar crops to many of us, things like cauliflower, broccoli, cabbage, turnips, etc. And these classic examples have really, I think, captured the imagination of many of us in brassica, ferns, and all sorts of other systems out there to understand how these species play a role in the landscape of plant diversity, as well as these days in the history of plant evolution as well. Aside from these sorts of really fun natural systems, there are many economically important polyploid and hybrid species, everything from potatoes to oats to bananas and kiwi fruits. You name it, there's almost certainly a polyploid or hybridization involved in the origins of many of our economically important plants. Everything from cotton to jeans, and even I like to point out this is a state record of fish here in California that is sort of cheating. It's a triploid hybrid with hybrid vigor. So, but there are these kinds of organisms are found throughout not only nature but have been important to the economic uses of these organisms. But across nature, if we go out and look on the landscape, based on our analyses and a few different papers over the years that we've done this in, you find that polyploid species make up roughly about 30%, 35% of flowering plant species outside. So basically, if you go outside and you look around, on average, 1 out of 3 species will be a contemporary polyploid species. And we've estimated in the past that about 15% or so of angiosperm speciation events and 30% or so of fern speciation events are associated with that ploidy change. So polyploidy is a substantial contributor to the diversity of land plants, as well as to the speciation processes of land plants. We can further divide polyploidy into broad categories. These are fuzzy, and there's a whole spectrum of this, but from allopolyploids to autopolyploids. And allopolyploids being polyploid species that arise via hybridization between 2 diploids or two different species. They don't have to be diploids necessarily. That gives rise to these allopolyploids. And autos are polyploids formed from self-duplication. And they're, in our survey at least, we find that they're roughly equal frequencies in nature. That many allos are described, many autos are undescribed as segregating autopolyploid cytotypes and populations. But if you look at the data, they look like, to us at least, they're about equal frequency, but that could be debated, I suppose. So there's a lot of allopolyploids and autopolyploids out there hiding out there on nature. These are all contemporary things, right? These are all species that are currently polyploid. But we know, in one of the great revolutions of the last 25 years, since that last IBC, I would argue, actually, is that as we've sequenced genomes, as we've looked at the history of what's happening inside those genomes, we see evidence for many rounds of ancient whole genome duplication throughout the history of plant evolution. I really love this figure. This is from the Vitis genome, partly because it shows these patterns so nice. The history of this, in this case, the eudicot hexaploidy. And you can see clearly that it shows a number of things, but one thing this paper shows nicely is it illustrates that genomes of flowering plants can have different levels of conservation of their synteny and different amounts of fractionation and rearrangement. And you can see that this ancient paleohexaploidy here, you can see there's sort of this nice orange gold color. There are 3 chromosomes still largely intact in Vitis, but these have been reduplicated and scrambled at least a couple of times here in Arabidopsis and again in Populus as well. And so even from these same, so plant genomes look like this mosaic, right, of layers of duplication on top of each other, and that are then fractionated and lost and turned over time. And this has left a legacy of paleopolyploidy then throughout plant genomes. With as many as up to maybe 2/3 of, for example, the genes in the soybean genome are paleologs derived from ancient polyploidy. Those genes were born in a duplication event, to something like down to 25% in a highly fractionated and diploidized genome like Arabidopsis, where even a quarter still of those paralogs and those genes in the genome are derived from that ancient polyploidy. And we call these paleologs, we call them onologs. There's different sorts of terms for these, but a substantial fraction of just raw genetic diversity in genes in plants, loci themselves, derived from these ancient polyploid events. And so I like to think that, perhaps one perspective that's changed since IBC in 1999, since my very first meeting, is that polyploidy has gone from being this sort of interesting, cute kind of thing to, in my opinion, really being the story of plant evolution. That plant evolution itself is a story of polyploid evolution. And as we'll see, all flowering plants have a polyploid ancestry. Everything's gone through diploidization, and so really understanding the biology of polyploidy and the biology of diploidization is fundamental to understanding the evolution of plant diversity, their genomes, and their genetics. And so I want to look at two questions today that we've been studying in my lab. One is a question that unites a bunch of different themes in our group, but it's really focused on reconciling these different perspectives on that we've gotten on whole genome duplication and polyploid speciation, which is that how do we reconcile multiple rounds of ancient polyploidy throughout the history of flowering plants with estimates that among contemporary polyploid species that they may have lower net diversification rates than many of their diploid relatives. How do we reconcile those two results? And then what are the impacts then of these ancient polyploidies that are in the history of flowering plants? What are the impacts of those duplication events on diversity and adaptation among contemporary plant species? And so as I said before, genome sequencing has revealed that there are many rounds of whole genome duplication throughout the history of flowering plants. And we've had all these glimpses for over the last 25 years, but really that first decade of the 2000s was making these new discoveries and, there's another one. Okay, this is getting interesting. How do we put them together, right? Where do they go? And in 2008, 2009, a group of us were organized with the 1KP project, the 1,000 Plant Transcriptome Initiative, and we engaged in this large genomic survey of green plants. I see many of you out there, so you all know what I'm talking about. But many of you are in the audience that helped us out with this and worked with us and were collaborators on this. And a part of that team, but we collected around 1,500 transcriptomes from across the phylogeny of green plants. And my lab led the analyses of paleopolyploidy across this tree that we developed in collaboration with the other 1KP members. And I was really excited about this question here. These questions, I suppose. When, where, and how many whole genome duplications are there in the plant tree of life? And we used a variety of tools, some of them newly developed for this task in my lab. And this was a project that was led by it was really a team effort led by Dr. Zhang Li, a former graduate student in my lab, who's just starting a faculty position at Miami University, like August 1, I believe, is the start date, as well as a few really outstanding undergrads, bioinformatics undergrads in our program at Arizona. But primarily Thomas Kidder here, who, excuse me, I just did something there. I got out of the menu. Let's see if I can pull that back. Well, okay, let's see. I apologize for pushing a long button there. Anyway, full screen, where do we see it? Oh, I'll let those guys do it. I apologize. If I can go backwards then. There we go. Apologies. I apologize for that. And Thomas here really led the analyses of this. This was really important for this. And here he is, what I call this our scroll of polyploidy. This is about a 30-meter long printout of the entire 1KP tree, and we were manually double checking and placing things before trying to put them all in the, verifying those locations among different analyses. And what we found in that analysis is that we inferred about 250, 245 ancient whole genome duplications throughout the flowering plant phylogeny across those 1,500 green plant species. And it really fun about this is that among flowering plant species, it was about 13% of the branches in that flowering plant phylogeny had an inferred whole genome duplication. So there's lots of ancient polyploidy distributed throughout the history of flowering plants. And we've recently reanalyzed all of these data with 6 different ways with updated tools since 2019. And this is coming out next month in a special issue of the American Journal of Botany that I'm leading on polyploidy. And this is the cover here. And this was work that was led by Michael McKibben. We ran a whole bunch of different species trees, a whole bunch of different analyses. The punchline is the results are basically identical to the 1KP. We get almost identical number of polyploidy inferred in the flowering plants, doesn't matter what method we use. They all end up basically in the same places on the same trees, but you change the species tree and you get different placements, as you might imagine. And so for these analyses of paleopolyploidy, one of the things that I just want to highlight that comes out of the study is that the phylogenomic methods are important, but they largely agree with each other, even though they use slightly different algorithms and different statistics. But species tree has a really big impact because all of these methods map those polyploidies onto that species tree. So we need to really think about how we build in uncertainty in species tree topologies into these methods, or just be cognizant of running multiple species trees in these kinds of analyses. But be sure to check that out. This is online now. This paper's been out for a while on bioRxiv, as well as on AJB's website as well. But keep an eye out. We got a great special issue, lots of papers. Some of you in the room are no doubt on there, and that's coming out next month. So if we look across, though, the whole phylogeny of flowering plants, and this is really kind of one of the things I was really excited about doing with the 1KP data, was getting this number here. We can then summarize how many rounds of polyploidy each species has had. So you just take the phylogeny, count back from the tip to the root, and say, how many rounds did this species have? All right, how many rounds of polyploidy did this species have? And go back and forth. We see that most flowering plants had 3 to 4 rounds of polyploidy. Oh, let me go back there, sorry. And they had 3 to 4 rounds of polyploidy in most flowering plant species. And this is nearly identical to, we got almost the same number in our more recent updated analysis with Michael McKibben's work here, with this nice distribution of 3.5 rounds of polyploidy on average in a flowering plant genome, same as the 1KP. And that's really interesting, right? I mean, it's cool, but when I see this number, the first thing I think is, okay, now I know the number, I want to know why. Why is it 3.5? Why isn't it 8? Why isn't it 1? What is special about this, and why did we end up at this particular number of rounds of polyploidy on average across so many different species of flowering plants? Why is this the distribution? And in particular, it's striking because our other work that we've done, and other folks as well, even though we see these many rounds of polyploidy throughout the tree, we also know that from contemporary estimates of diversification rates of polyploid species, if you go outside and look within genera, look at the ongoing diversification rate estimates from tools like BISI or HISI or other ways of assessing speciation and extinction rates, you see that polyploid plants tend to have lower net diversification rates than their diploid relatives out there in the landscape. So how do you reconcile these two seemingly opposing results, right? If this contemporary polyploid diversification is so low, if so many of them don't actually go on to do much, how can there be so many ancient whole genome duplications in the history of flowering plants? And so that's what we're going to look at today. And so there are some possible explanations. Perhaps our estimates are wrong, right? That's one explanation. Perhaps our inferences of diversification rates are wrong. Perhaps we don't have a good handle on those. Perhaps we've inferred too many ancient whole genome duplications, so perhaps our inferences of ancient polyploidy are wrong. But at the same time, we might have inferred too many of those. Other folks argue that we haven't found enough, that there's more out there you're clearly probably missing. But these are probably, we'll see later on, not necessarily accurate. I think we're actually probably doing a pretty good job as a field, right, in both our phylogenetic analyses of diversification rates and polyploidy. And our own, there's many analyses that have very similar results, though. So that's one possible explanation for this disparity. It may also be that polyploids, right, have had higher diversification rates in the past, right? That this is not a continual, that we don't have a constant, just average diversification rate among polyploid species. And I think, that totally seems reasonable. There's this potential, for example, this potential wave of whole genome duplications at the KPG boundary. And even if that's looser, or we put a lot more duplications in that tree, they don't necessarily cluster there quite so tightly. But even though that's not the case, it's easy to think and easy to imagine, right, as an evolutionary biologist, that diversification rates have likely varied over time, that in different environmental conditions, there may be traits associated with polyploidy that make it more advantageous or more disadvantageous at a different point in time. And so there may be these heterogeneity in those diversification rates that we just don't capture when we look outside on the landscape today. And as I said, it may also be that polyploids have diversification rates vary with other traits. And we know this is true. A couple of different studies, one from Rosanna Zeno-Ferguson and colleagues in the Solanaceae and one from our group in Nebraska KCE, led by Christian Roman Palacios, we see, that there's other traits associated with polyploidy can influence the diversification rate among these contemporary polyploids. And I'll go through an example from our work with Christian here real quick. And in this analysis, we analyzed a little bit over half of the contemporary diversity of Nebraska KC. We reconstructed phylogeny and reconstructed chromosomal evolution across the family in a couple of different ways, shown here in this really beautiful figure that Christian produced of the rates of chromosomal evolution across the family. And when we look at the diversification rate of polyploids and diploid species, we see here this is a plot of a Hissy model. This is a hidden speciation, a hidden state speciation and extinction model. And what this allows us to do is to ask whether there are potentially sort of more complexity, more structure within the data, that there may be some trait that we're not accounting for, where there are some polyploids or some diploids that have really different diversification rates, and that those may be then associated with a hidden state. And Hissy found that, yes, that seems to be the case in Nebraska KCE. So if you just look at the overall average of diploid speciation and extinction rates, you see here that diploids, just like our general averages, diploid species have slightly higher net diversification rates than polyploid species here in this plot. But if you run this through Hissy and look for evidence of this hidden structure, you'll see that if you're a hidden state B, for example, and you're a polyploid, the polyploids are here in the red, you're not doing so hot compared to the hidden state B diploids, which have a really fantastic advantage over everybody else, right? But hidden state A, the polyploids are doing slightly better than diploids with hidden state A. Now, the fun part about this is we don't know what hidden state is in this analysis. It may be something we think in follow-up analyses that we've done this past year with mating systems. It may be something to do with annuality and perenniality, but to be determined still what that hidden state is. But you can see that there's this complexity then of the interaction of ploidy and traits that can give rise to complexity in these relationships, even though the average overall looks something like this, where diploids have a slightly higher net diversification rate than their polyploid relatives. So a final option is that there is no conflict, right, among these different estimates, that our diversification rate estimates are reasonably good, our estimates of ancient polyploidy are reasonably good, and that these all largely agree, right? And that's what we're going to explore, and I'm going to show you some results of our exploration of that today, using a series of simulations and modeling to test if this inferred number of whole genome duplications is consistent with these estimated net diversification rates. To do this, we modified a version of BISSI. So this is the binary state speciation and extinction model. And this was work led by Xing Zhan and assisted by Chouyu Jiang in the lab. And the way this works is that if you want to think about this BISSI model, we have a pool of diploids and a pool of polyploid species, and each of them have their own speciation and their extinction rates, as well as this transition rates then between those two states is polyploidization and diploidization. And so how we do this is that we run simulations under a variety of parameters, and we count then the number of whole genome duplications that accumulate throughout each of these simulated trees, these thousands and thousands of simulated trees. And it looks something like this. We simulate these trees, and we can then say, for diploid species and for each species in the tip, we can then count these transitions that happen, and these are just color-coded in this yellow and gold color here and blue. And then we can count those transitions just like we do when we look at that empirical distribution of whole genome duplications across time. And so we can then parameterize this model using just random parameters. We can also parameterize it using empirical data. And so for just an exploratory sort of data set to look at how these phylogenetic dynamics work, right? We're using a set of arbitrary net diversification rates where the rate of diversification, for example, for polyploids is much, much higher than those for diploids, to where they're equal, to where diploids have much higher diversification rates than polyploid species. And we could set the polyploidization rate just for this analysis to say 0.15, which is the empirical estimate from our Wood et al. Paper back in 2009. Now, diploidization rate isn't exactly quantified in the same way, right? We don't have the same kinds of quantification for how fast things diploidize. We have a good sense of that they're fast or slow, but we don't have this sort of empirical number. So we can plug in that transition rate across a range of values. In this case, 0.2, 0.35, 0.65 to 0.8. So slow to fast rates of diploidization. And just sort of see how these work and to show you what this looks like, we do this for 1,000 taxa with 1,000 replications of each combination. And we get a plot that looks like this. All right, so what we're seeing here, I'll look at one example here. In each of these plots, we have a heat map of the number of rounds of polyploidy that occur in each lineage in these simulated trees. So the y-axis is that number of rounds, so we can see that under this set of conditions, we had one round of whole genome duplication up to 6 rounds of whole genome duplication. And on the x-axis, we have the diploidization rate, so from slow diploidization to fast diploidization. And each cell then, or each square here, is a different combination of diversification rates. So in this particular center cell, we've got the diversification rates of polyploids and diploid species are equal to each other. In the top right corner, polyploids have much higher extinction rates. They have a much lower net diversification rate compared to the diploids. And the bottom left corner, it's the opposite. Diploids have a much higher or much lower diversification rate compared to the polyploid species. So what do we see? Well, one of the fun things you can see across this is you can generally see there's a diagonal line from left to right. So you get more ancient whole genome duplications in these phylogenies when you have higher rates of diploidization. So going up but then back down from polyploid back to diploid leads to more rounds of polyploidy because you diploidize and then can polyploidize again. We also see it under conditions where polyploids have a lower net diversification rate than diploids. So you're essentially clearing the polyploids off the table either by diploidization or extinction, and then the diploidized paleopolyploids re-polyploidize again. This is similar to the dynamic we see in flowering plants, and that's what we see up here in this top right corner, where these have the diversification rate dynamics that are most similar to flowering plant species from our contemporary estimates of diversification rates. And what's interesting about this, even in this small set of simulations, we get 3 to 4 median whole genome duplications at the higher rates of diploidization, which is eerily what we saw in our empirical data for flowering plants. Now, what's also cool about this is we can ask the opposite question. What sorts of conditions in these models lead to fewer ancient whole genome duplications throughout a phylogeny? And those are conditions with lower rates of diploidization. So staying polyploid for a very long time, having this very slow rate of diploidization leads to fewer rounds of polyploidy ultimately as you persist in that polyploid state, as well as a situation where polyploid species have a net diversification rate greater than or equal to diploids. And this is pretty similar to what we think we see in fishes. Fishes have 1, maybe 2 rounds of polyploidy on average, but famously slow rates of diploidization. So you can look at the salmon genomes, a lot of these trout genomes, for example. They're 100 billion years old and they still have some multi-somic inheritance, right? They're still pretty tetraploid even after all that time. It's not just about maintaining the synteny and things, but they actually still seem to have loci that are segregating in a polyploid manner in some of these lineages. So this may actually be similar to what we see in the fishes. They've stuck around as diploids or as polyploids forever, really slow diploidization. All right. So that's fun. That's just playing with the parameters, and to give you a feel for how these work. What does this look like with empirical data? Right? We have empirical estimates of net diversification rates from a number of studies. So let's just take one from our work with Christian, looking at the Brassicaceae. And we can take those diversification rates from there. We can use, again, we don't know the diploidization rate, but we know it's pretty fast. But what does that mean exactly? But we can then simulate across a range of diploidization rates and use 4,000 taxa with 1,000 replications, so something similar to the size of our analyses in 2020. We get a plot that looks like this, a similar sort of heat map plotted a little differently. Across here on the x-axis again is the transition rate. Excuse me, we're trying to get the the pointer down there. The transition rate from polyploid back to diploid. So on the left side over here is the fast on this side, and on the other side, okay, is slow. And I went fast again. And what we see is that this general trend again, that as you have faster diploidization, you get more rounds of polyploidy. And if we look at our genomic analyses in the Brassicaceae, we estimate there's been about 1.8 average rounds of whole genome duplication since the Brassicaceae evolved in your average Brassicaceae. How's that? And that's basically where the core of our simulations ends up at in these analyses. And indeed, if you plot it in this fashion to summarize all of those heat maps, When you have a high rate of diploidization, we actually get the exact empirical estimate in this 1.7 to 1.8 range of rounds of polyploidy throughout the brassicaceae. And that matches our empirical analyses, which is, to me, is really, really exciting. We can also go back and look at our the Mayres et al. Paper from 2011 and say, let's take the averages of those net diversification rates. There were, I think we analyzed 40 to 45 genera in that analysis, ferns and angiosperms. And we can take those estimations, the range of net diversification rates from that study and put that into a similar set of simulations. We can use a range of polyploidization rates as well, using 0.15, for example, from Wood et al., and then bracketing that with 0.3, 0.45 from our other studies, our other estimates of the frequency of polyploidization, as well as look at the diploidization rate across the whole range of values. And in this case, we're doing 100,000 tacks in these simulations with 100 replications. It takes a long time to run these. So trying to do this at a scale of all flowering plants just takes, it's really long. These take a few weeks each to run, so to put it in perspective. And there's been some attempts to get this to go a little faster, but it's a little tricky. But anyway, things keep going extinct, and then you have to make new lineages, the way it works. So when you have 100,000 taxa, though, what does this look like? And so this is sort of thinking, what is this? This should be a proxy for what we see across all flowering plants. And we see something that looks like this. Again, that same QPD is the diploidization rate along the x-axis. So over there at 0.9 is relatively fast diploidization to slow diploidization over here. And again, remember from our 1KP and our recent McKibben et al. Analyses, flowering plants have an average of 3 to 4, 3.5 rounds of whole genome duplication. And what you see is that right there in this core thing, when we have in the core range of that distribution, when we have high rates of diploidization with those Mayres et al. Estimated no diversification rates from contemporary species, we get that 3 to 4 rounds of polyploidy on average, right? And so those values that we're producing, so we're able to take those contemporary net diversification rate estimates and with high rates of diploidization, which we think is how this works in most flowering plant species, get the number of whole genome duplications that we observe in our genomic analyses. And so this is taking data from phylogenies and combining this with these whole genome analyses, and we can see that same distribution. And so if we look at this here in a different kind of plot where we can summarize these into a larger, different kind of heat map, we see right there at that estimated rate of polyploid speciation, about 30% of polyploids, speciation events being polyploidy or so, we get that nice 3.6, 3.9 rounds of whole genome duplication at these higher rates of diploidization in our simulations. So what do these simulations tell us about the evolution of plant diversity and plant genome complexity and polyploid speciation, right? Well, one of the things that I think they tell us is that these higher diploidization rates that we do see in flowering plants are important, and they're really important because they contribute to more rounds of whole genome duplication. That's one thing that they're doing in this phylogenetic dynamic here. And in particular, we also see that there are more rounds of whole genome duplication in the history of flowering plants when polyploid species have lower net diversification rates among contemporary polyploidy, at least, have lower net diversification rates than their diploid relatives. And that I think this is exciting because the simulations here, right, we used contemporary net diversification rate estimates, and they're producing numbers of whole genome duplications in our phylogenomic analyses that are consistent with those observations, which to me suggests that, perhaps this is multiple different sort of lines of evidence that we're sort of confirming, and that maybe our ability to infer both net diversification rates, to infer these phylogenetic diversification rates, and our ability to infer genome duplications in those numbers, we may not be doing too bad, right? These simulations suggest that we may be in the right ballpark. That we're not necessarily too far off on those diversification rate estimates, and we're not necessarily too far off on the number of rounds of polyploidy that we infer. And that diploidization itself may be an underappreciated driver of ancient whole genome duplication numbers across the phylogeny, not just in plants, but across eukaryotes in general. We see this same sort of dynamic seems to predict what's happening in fish genomes, but as well as in flowering plant genomes at two different ends of the spectra of polyploidization and diversification rates. And so if, among flowering plants at least, if contemporary polyploid diversification rates are kind of low compared to diploids or slightly below them, How can there be so many ancient whole genome duplications? And it may just be that a big part of this is that dynamic with fast diploidization that we see across the flowering plants. So what's next? Well, one thing that would be fun to do is to track multiple states of polyploidy in these models. Right now we're just doing diploid to polyploid cycles. And so we can capture those transitions. I will say that I think we're capturing most of the biology now. We've got other analyses ongoing and papers that are out there on, inferring the frequency of paleohexaploidy, paleooctoploidy, et cetera. And most of them are going tetraploid back down again, right? Most of them are just doing paleotetraploid back and then back to diploidization before you get these higher ploidy level polyploid events throughout the history of trees. So I think we're capturing a lot of them, but it would be nice to be able to include that dynamic in a future version of this model. But this is a really fun one. One of the great things about doing simulations and modeling is that it helps you sort of sharpen the kinds of things you should focus on empirically. And I think one of the things that this suggests that is really important is diploidization, right? And what is diploidization? Well, and how do we study that? And we've been thinking about this a lot for the last few years. But what exactly is diploidization? Well, in this model, it's when species return to a diploid diversification rate. What does that mean empirically in the real world? I don't know. Diploidization itself, as I'll show you, is complicated. There are many. I'd like to write a new review on this. We wrote a review a few years ago, but there's sort of different ways you can think about diploidization, and I'll walk through the different examples. But there are different parameters that make up diploidization. It's not just one thing exactly. And so what part of that diploidization actually means you get back, in this case, to a diploid diversification rate is going to, I think, vary among lineages potentially, but we certainly don't have an understanding of that right now. And I think that's really interesting research to be done, and I think novel sort of experimental work to be designed and analytical work to be designed as well to get at that particular question. And we're trying to do that in our lab a little bit, working, for example, with Song-Yen Kang and Michael McKibben in the lab. We're trying to look at this variation in diploidization processes and outcomes across the phylogeny. We published an annual review a few years ago now. It was the summer of 2020, we're finishing it up. I don't even remember writing half of this, to be honest, after that pandemic year. But on these patterns and processes of diploidization. And we could see, for example, in that analysis, that there's a lot of variation in diploidization processes and rates of fractionation across the phylogeny, lots of variation in rates of chromosomal evolution and rates of chromosomal evolution across the phylogeny, where I think we can really begin to dig into the different consequences of diploidization itself and the mechanisms driving that, and what gets us back, for example, to a diploid diversification rate, thinking about it for that model's perspective. And this did not render. Oh. We also see that there are variation in chromosome numbers. This is work by Christian Roman Palacios. We've been working on this. We built an animal chromosome count database to complement our plant chromosome, the plant chromosome count database. And what you would see here, if you could, is that flowering plants and animals have the exact same chromosome number distribution. And if I took away the color coding, I don't think any of us would be able to recognize which lineage is which. And so digging into these greater questions too about what drives chromosomal evolution and chromosome number evolution, and check out Christian's paper in the Journal of Evolutionary Biology if you want to see this really nice figure that unfortunately didn't render here. As well as looking at other lineages, so looking at different extremes of these lineages, work by Sylvia Canosian, who was unable to present her talk on Monday because of flight troubles, on genome evolution in Homosperous ferns. And one of those questions that's been near and dear to my heart for a long time is understanding those high chromosome numbers that the Homosperous lineages exhibit. Sylvia's got some exciting work on that she'll be showing soon, as well as species at the other extreme. And in this case, we've got a whole line of work that Jeff Finch, a student in the lab, is working on in Xanthusma, in the Macrantherinae, a clade of the compositae, looking at a lineage that is really exciting with 2 chromosomes here. We've got a great genome from this. This is a genome from my neighbor's front yard. This grows outside, so it seems like I should work on it, this species with only 2 chromosomes. And you can see here, we've got these one chromosome here with a centromere not assembled. And then because these things are fused, you've got these sort of recent centromeres in there lined up against the lettuce genome in this assembly. But the genomes are the same size. They just smashed all of them together. So looking at the population dynamics and the evolutionary drivers of chromosome number reduction and chromosome evolution in general is really exciting as a part of this work to understand what's going on with diploidization. And I want to just talk a little bit here at the end, a little bit about a slightly different vignette that it highlights that the important biology, though, that happens in these polyploid and paleopolyploid lineages is not confined to the period when species are polyploid. I want to show an example of some of our ongoing work looking at the legacy of paleopolyploidy in flowering plants with an example from the brassicas and some crops that we've been studying, where we see that there's higher genetic diversity and domestication associated with these diploidized polyploid lineages, and that paleopolyploidy has been really important for the food we eat and adaptation and evolution of plants in general. And just a very quick little look at the story. This is work that was led by Dr. Jingshuai Qi in the lab a few years ago now, and we worked with Chris Piers and other colleagues to sequence a number of Brassica rapa accessions to study ultimately asking this question about how ancient polyploidy in the Brassicaceae is associated with the diversification of these crops and the diversity of the Brassica crops. And we sequenced 126 new accessions and mapped 30-some thousand SNPs back to the reference. And I always like to sort of point out that we did one of the first demographic analyses of the history of Brassica domestication. And I always like to point out here that we were able to corroborate some of these domestication events with the primary literature here on the left. Some ancient Chinese poetry that mentions old Brassica rapa crops, as well as this tablet that records their history in Babylon, which I always think is great fun. And we were able to get dates that corroborated with this historical records of Brassica crops. But the polyploid story with this to me is really exciting and really cool, which is that when we look at the candidate domestication genes associated with the domestication of these Brassica rapa crops, in this case, through a variety of different analyses, we see that the candidate genes are enriched for paleologs. So here gold are paleologs and non-paleologs are in blue, and this little asterisk indicates there's a significant enrichment. So genes derived from that ancient Brassica hexaploidy, in this case, the Brassicaceae hexaploidization, are enriched among the candidate domestication genes in the Brassica rapa crops. And it doesn't matter if you're differentially expressed, as well as genes that are from our literature survey, looking at genes that have been fine mapped in other analyses. And we can classify the paleologs in different ways. So like, how do you know that this is a gene from ancient polyploidy versus not? And we see the same pattern if you use different methods in this particular case. And this is a paper that we published a few years ago in New Phytologists. So check that out if you'd like to read more about that and all the various approaches we use to make these inferences. But one of the fun things about this too is that when we looked at the genes, we also saw that these paleologs had higher diversity at both synonymous and non-synonymous sites. So this is consistent with this idea that perhaps these genes that are being maintained in duplicate from a long time from these ancient polyploidies might be buffering each other, might have a little bit of relaxed selection associated with them that leads to this elevated diversity that may then be really important later. And we saw this here in the brassicas. There was a strong evidence that the paleologs had this pattern of molecular evolution consistent with relaxed selection. And this raises the question then, how important then are these paleologs for domestication in general? How important is this extra variation that these fractions of plant genomes, of diploidized plant genomes, how important is that variation for adaptation, or in this case, domestication? And so we did a follow-up study here with again, Michael McKibben. You've seen a lot of Michael's work in this talk. We developed a new machine learning algorithm to rapidly classify paleologs. For those of you that have used some of the more sophisticated paleolog classification tools, they can be quite computationally intense and take many months to run. And so we worked on developing an approach that we call Fractify that we published a couple years ago or last year to classify these paleologs and the fractionation sections of plant genomes. And this basically takes, about 28 features or so from synteny, phylogenetic data, and other features of plant genomes to classify these paleologs. Things like synteny block features, KS values, et cetera, that are all baked into this model. And it can pretty well classify non-paleologs from paleologs that in this particular version of the model that's trained up to classify them as genes that are retained in single copy, double copy, or triple copy, so that we can analyze hexaploid genomes as well as tetraploid genomes in this case. And it does a pretty good job at doing this. And it does a pretty good job still, even with when there's been many rounds of polyploidy, as we know. It can get a little bit of a blind spot if there are too many stacked up. I'm trying to get this pointer to look at the right spot. But down there on the left in the single retain, you can see that there's sometimes it does a poor job right at the very front of the peaks, but then if they're too stacked up on each other, but overall it does pretty well. So we use this to classify paralogs in 22 domesticated species, including things like brassicas, corn, apples, cotton, rice, and many more. And these are across a variety of different kinds of agronomic traits, selection on leaf and root size, oil production, or fruit size. And these included 15 different whole genome duplication events in the history of these crops, 7 unique and that 8 were shared, and across time from about 5 million years to 117 million years. And what we see is this. This is a little big wall to look at. The important thing to focus on are these asterisks on the side. These are cases where we had a significant enrichment of paleologs among the candidate genes from these studies. So these are candidate gene lists that come from the studies themselves and with our paleolog classifications on top. And what we saw is that to summarize that whole distribution into a much easier to digest plot is that paleologs appear to be important in the domestication of many crops. So this is not just a story in brassica, it's across all flowering plant crops as far as we could tell. And what's really exciting about this too is that the single retain genes in particular, so genes that have diploidized back down to a single copy or have retained in a double copy, are most often involved in these domestication lists. They're highly enriched. And these genes that are retained in triple copy are generally not as much. And we think this might make sense based on sort of the dosage balance ideas. You've got strong purifying selection to maintain genes that are potentially that are being maintained in triplicate after hexaploidy or in duplicate after tetraploidy. But maybe perhaps they're less constrained if they got this little bounty of variation from being in a relaxed selection state for a while and then went back to being diploidized. They may be less constrained to be involved and recruited in domestication. Now, you might say, oh, maybe this is just paralogs in general, right? What's special about polyploid paralogs? Well, they're the only ones that are significantly enriched in the candidate gene list. We can look at every other paralog in these genomes, and none of them, no other category is enriched. This is in particular a classification by MC scan of different paralog types, dispersed, proximal, segmental, tandem duplicates. They're all over the place, but none of them are significantly enriched in candidate gene list. Only genes from ancient polyploidies are. And this is consistent, right, with our analysis in Brassica rapa. So this is a story that isn't just a Brassica story, but is a story of the domestication of plants in general, that ancient polyploidy and variation from ancient polyploidy has been important for the evolution of, for the domestication of crops, and likely for the evolution of plants in general. And so what I think is really exciting about this is that even after millions of years, polyploidy is still influencing genetic variation in plants, right? So these are events that happened many millions of years ago. They're diploidized species, but that structure, that genome, the genomic changes and that legacy of those ancient polyploid events has left this long shadow on plant evolution and plant genomic diversity. And I think this is really fun to think about, we're cross scales here with our macroevolutionary analyses of polyploidy and where duplication events have happened, and that all directly connects in with our analyses of contemporary plant species, contemporary plant population genetics. But I think really links those different scales of evolution as well. All right, so a few take-home messages as I wrap up here. There's a lot of paleopolyploidy in the history of land plants. I didn't include this actually in my little back of the envelope calculation, but based on those discovery rates, maybe there's about 8,000 more ancient whole genome duplications to discover if we were to theoretically sample every species of land plants. We might see about another 8 grand or so. It could be an order of magnitude off, but don't hold me to it. But there's a lot out there. And that what we see, at least in our simulation studies, is that this combination of rapid diploidization combined with these low average contemporary net diversification rates of polyploids explains in our simulation framework these multiple rounds of whole genome duplications, and it's consistent with our empirical observations. This is what to me is really exciting, that we reproduce those empirical observations in our simulations. And remnants of these ancient whole genome duplications have been important for contemporary plant evolution and adaptation. They harbor elevated genetic diversity. I didn't get into that too much in this talk, but go read our paper. There's a couple other papers on the way too. these are the regions of the genome and flowering plant genomes that have more genetic diversity than other parts. And for that reason, I believe, and we're testing this, these are regions then that are enriched among candidate genes associated with crop domestication. And if we go out and look in our natural species, our natural systems, at natural selection and adaptation to the environment, I would expect the same thing. So this legacy of paleopolyploidy is immense on flowering plant evolution. And I think that it really highlights that studying polyploidy and diploidization is critical to understanding the evolution of plants. The story of plant evolution is a story of polyploid evolution. Even in the diploids, you're still can't get out of the shadow of polyploidy and its impacts on what's happening in those species. And before I wrap up, finally, I want to highlight that it will be literally 1 year from today, on July 26, 2025, we'll be hosting Botany again in Tucson, Botany 2025. We had it in 2019. If you like this temperature, we'll have the same weather, except we'll have some monsoon storms. We call it our season of lightning and rainbows. Here's a little rainbow from a monsoon storm there last year, the last time in 2019. But come join us in Tucson for Botany 2025. I know that they're getting the logo ready next week. Otherwise, I'd have a more official looking logo. But please join us again. It was really wonderful hosting everyone in Tucson, and I look forward to doing that again. And for those of you that are participating in the Polyploid webinar, look out for emails. I was going to use this as a launching point to start the fall 20 cycle. If you want to join us, feel free to connect with me here at the meeting, and we'll get you on the list. But otherwise, I'd like to thank everyone in my current lab, funding agencies that have funded us over time. We just had a new training grant in NRT that I'm leading called Cambium that was funded this week. So if you're a US citizen, student, and are interested in joining our lab to work on these questions of polyploidy and climate change and adaptation, we have new funding to support that work. Come see us. But otherwise, I'm happy to take any questions. Thank you.
Thank you very much, Mike, for this excellent lecture. Please, it's open to question.
Thanks. Well, you mentioned that the diversification rates in diploids are higher now than in polyploids. What about allopolyploids and autopolyploids? Are there significant differences too?
That's a really great question, and one I've been chasing for years. I will just say that in the maybe it's very difficult. We've tried to build some new frameworks to be able to do that in a more sophisticated way. That was part of the work back in 2016. It's hard because a lot of the autos are undescribed cytotypes. So when you sort of think about classifying diploid speciation rates in a lot of the phylogenies, there's hidden autos that confound the analysis. And we were trying to get around that problem by really figuring out a better way to define them. So I don't have an answer. My instincts say that we have a new analysis, new tool called All-Out, Allo Auto, but All-Out that a student in my lab has developed, Alex Stewart, a first-year student. My instincts are that a lot of the to infer ancient allo versus ancient auto, and my instincts are that probably a lot of these ancient ones that have gone on to do really well are allos, but that's to be determined. So, how's that? Hexaploids, paleohexaploids also do really well. We have a paper on that coming out from a new method for inferring paleohexaploids and rapidly classifying them. So, and that may be because a lot of them are allos, or maybe that extra structural and that extra variation, extra genes means something, but lots of reasons for that. Yeah.
Okay. Thanks.
Thank you. Bye.
Thank you very much. A really exciting talk. Really enjoyed that. I wondered whether you had any comments on the evolution of high ploidy numbers in flowering plants. Do you think the most likely evolutionary paths are through recurrent auto or allo or hybridization between species with already high ploidy levels?
That's a really good question. You know, you can get these like nonaploids and really high ploidal level systems, and I recently used one of those we have a new key code on the outside of the lab, and you have to be a polyploid nerd to know what the ploidy level of that species is to get the combination. But anyway, yeah, you know, it seems like in a lot of those high cases that it's some combination. Often they get up to a high base number, right, with auto rounds, and then you get hybridization among a couple of those lineages that gets you into that stratosphere of contemporary polyploid levels, which means for sequencing and analyzing them, I mean, I think this is also, it's kind of a nightmare to disentangle them. So in these cases, but they most often, at least based on my understanding and the examples I've looked at, have a lot of auto polyploidy, but it's a combination of those typically, or at least they're really, you know, high segmental allos or something that's really hard to disentangle. Are those, you know, from the karyotypic information at least, are those subgenomes different or not? So yeah. Yeah, Pat. I see you, Pat.
Hello, thank you. Okay.
Oh, sorry.
Is it me, Andrew Leach? Hello. Mike, very nice talk. Thank you, very interesting. You putting a lot of weight on rates of diploidization. What exactly do you mean by that in sort of millions of years and genes, or what does it actually mean, 0.9, whatever?
Absolutely.
Number?
That's a really good question. And in the model, that's what I mean. We don't exactly know what this means. It's the rate in this case at which, very specifically in this context, at which these lineages go back to having a diploid speciation and extinction rates. So it's that transition back to that state. Whether that is, you know, because they've diploidized, what the biology of that is that because they've, you know, species have gone back from multisomic or tetrasomic inheritance, for example, back to disomic inheritance, or lost so many chromosomes, or whatever that might be, is a really good question. But it does tell us that those numbers give us a sense of the rapidity or the fraction, you know, how fast those are going each generation in the model. So at the higher numbers, many more lineages in the model are going back to diploid, so it's faster diploidization rates. Yeah, putting it in, these aren't necessarily in terms of millions of years in this case, so this is units of the tree, because these aren't calibrated in that way. But you could think of them that way if you wanted, if you were to think of it as a dated tree. But yeah, it's that salient biology that you know, we need to understand. But the very specific thing to keep in mind is in this particular model, it's when they go back to having a diploid speciation and extinction rate. And what that biology is a different question. Yep. Okay, I'll Pat now. Yeah. Sorry, I couldn't see.
Mike, great talk. Can you tell us what you'll be telling us in IBC C in 6,000 years?
It's a good question. If I last that long. Well, there will be, you know, a wonderful, I should plug it, Jacques Monod conference next fall in Roscoe, France, on polyploidy that I'll be speaking at, and I'll likely be talking about some work that is ongoing right now that should be done, hopefully, looking at testing this relationship between diploidization and diversification, and whether we see that these are, you know, does diversification precede diploidization, or does diploidization precede diversification of these ancient polyploids? And we've got some really fun work on that and some new models that we're trying to develop to simulate genome synteny and structure to really test those hypotheses. So.
The particularly important thing is the stress that we're putting on the world's flora going to increase the benefits of polyploidy?
So, yeah, what? So, well, I have a paper that we're writing right now, and I'll just say that it looks like polyploids are doing much better. If you look at all the data we have on species extinctions and things like this among contemporary species, polyploids are doing way better than diploids. And so we know from our ecological work and other folks' work too, right, that they do seem to do better. And they have broader niches than their diploid relatives. And this extra variation that they may have, this flexibility and stress may give them a significant advantage. And I think we empirically see that. So keep your eyes out for that paper this fall. But it's a pretty significant advantage in biology terms, so that we're seeing in the data. So. autopolyploid cytotypes missing, not included, which confound many of the analyses of contemporary polyploidy. But anyway.
I think maybe one last question, please. Anyone? Yeah.
All right.
Hi, Mike. You showed those nice graphs with differences in polyploidization rates in gymnosperms, ferns, angiosperms. So within angiosperms, the anagrade taxa are diversifying very differently from the mesangiosperms, and I wonder if there's a polyploidization effect there.
Yeah, there is, and I think that it's related to woodiness. So we're looking at some work where we've got a student working on this right now, looking at Rosanna Zindel-Ferguson had this great paper back in 2017, and Rosanna and I've talked about this a couple of times now. We've got a student working on this where we're looking at those paleopolyploidies and the reconstructions of ancestral woodiness across the history, and I think that's potentially what's happening there. There's just potentially less polyploidy in that anagrade because of so many of them are woody, right? And there's a strong ancestral woodiness there. But yeah, that's also an important point. Like, the evolution of herbaceousness is really, like, the number of rounds of polyploidy go up. So there's a lot of stuff happening at that same time too. So those things where you see like 7 or 8 rounds of whole genome duplication are all basically herbaceous rosids or sterids, and they're just kind of clicking along though. And I think maybe some of it's just their evolutions. They're moving faster, right? In some ways on these cycles, I think is what's happening. So, all right.
Okay, thank you very much, and thanks, Mike, for this excellent.
Thank you so much, everybody. Appreciate it.